In this video, we will discuss about GlyTR1 and GlyTR2 toxicity on mouse with human-like target density. We will discuss what does this human-like target density means when we reach to this panel E. Let us first start with the panel A, where they compare the staining of the different organs between the humans and the mouse. So first, and what they are able to find out is that when they check it for the L-PHA lectin, they are able to see that most of the organs between of humans and mouse behave similarly, except for the pancreas, which got stained with L-PHA lectin in the case of human, but there is no staining in the case of mouse. And when they check it for the GlyTR2, in GlyTR2 they do not see that stark difference. However, they are able to see that in both human and mouse behave similarly, and in the case of stomach we can see the more bindings can be seen. And thereafter, once validating that the humans and mouse organs behave similarly for their GlyTR2 and also for the L-PHA lectin, then what they did is they inject this GlyTR1 in the mouse and check whether it got accumulated within which part of the body. And what they are able to find out after 8 hours, they are able to see that most of this GlyTR1 is accumulated in the liver, and to some extent it is in the spleen, and which is consistent with the literature, as most of the clearing proteins, they are usually accumulated in the liver. This is what they have find out, and these results are same for the GlyTR1 and also for the GlyTR2. Quantifications are shown in the panel C and D, where we can see that except for the liver, the amount of the GlyTR1 and GlyTR2 CAR-T are very low in the other parts of the organ. So once validating that it is not accumulated within the body, next they wanted to check with this human-like target density. So in that case, what they did is they took the mouse, and in the mouse, in order to avoid immunogenic reactions, they knocked down this MSC1 and MSC2, and or the mouse does not have MSC1 and MSC2, and they inject these PBMCs on day 0 and check the PBMC engraftment at day 14, and at the day 15 they again inject these PBMCs, and thereafter at day 16 they treat the mice with 3 different conditions. One is for the control, that is with PBS, and in the other case they treat it with this GlyTR1 and GlyTR2 with a 10 microgram and at 2 different time intervals. Then after day 28 they euthanize and analyze the mice, and first they check the body weight. So what they are able to find, there is no significant changes in the body weight between the control and the GlyTR1 and GlyTR2 conditions. And next they check the accumulations of the or we can say pathology scores of different organs, whether it got different changes because of the presence of GlyTR1 and GlyTR2 or not. And what they are able to see that for the kidney, stomach, they can see the in all the 3 conditions their organs behave similarly. And even for the small intestine, duodenum and jejunum, and also lumen, in that case the presence of this GlyTR1 GlyTR2 PBS is almost negligible, or we can say the pathological scores are 0. So indicating that this GlyTR1 and GlyTR2 does not show any toxicity when they check it when they check the accumulations of both of this CAR-T construct, and also it does not show any changes when they use the PBMC cells into the mice. So this in all together supports their point that GlyTR1 and GlyTR2 are safe to be used on the patients who are affected with the cancers. So I hope you understood their experiments, how they conducted, what is the purpose of the experiments, that is to check the toxicity effect of GlyTR1 and GlyTR2. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.